So, welcome back. Today, I'm going to be discussing the next part of our cellular energetics unit, and I'm going to be focusing on the processes of photosynthesis. And so in the last video, I started by thinking I was going to talk about cellular respiration and photosynthesis in the last video. And then I said, I'll just talk about cellular respiration and photosynthesis in the next video. And then I decided, you know what? I'm going to talk about photosynthesis and cellular respiration in their own separate videos. So that'll give me a little bit more time to explore the mechanisms of each of the overall processes. And then at the end of one of the videos, or maybe I'll get some video. No, definitely at the end of the cellular respiration video, I will do a little section on how they tie into one another. Right. So for now, I'm just going to talk about photosynthesis. Right? It's a notion that I'm sure you're familiar with. We have cute, adorable plants catching up the energy from the sunlight and turning that energy into sugar. And now that, of course, is a terrible way to say it because the sunlight does not turn into sugar. We're using the free energy from the sunlight to create sugar. The energy is doing the work. The sugar is created from matter. So we're going to discuss how plants achieve this amazing feat of storing a non-material energy thingy inside of a material matter piece like sugar. Cool? Can't wait. So I'm going to begin with the anatomy of photosynthesis. And start with the structures involved in photosynthesis. And so now, of course, the first type of organism that comes to mind when we speak of photosynthesis is plants. Those big, beautiful eukaryotic creatures that surround us in our everyday life. We owe our existence to those beautiful organisms. And so plants utilize a specialized organelle inside of their cells called a chloroplast. The chloroplast is the structure that is functionally responsible for the process of photosynthesis as an overall chemical reaction. Now, plants are not the only organisms that can do photosynthesis. So we have advanced algae that do photosynthesis that utilize chloroplast as well. But we also have bacteria who are prokaryotic, who are able to do photosynthesis. And so clearly, the prokaryotic bacteria do not possess chloroplast because chloroplasts are membrane-bound organelles. And prokaryotes don't have those. So instead of having a chloroplast, photosynthetic bacteria have a highly folded cellular membrane, and they have these regions of folds that function in the same way that the chloroplasts do. Okay? So it's important to keep in mind that there are prokaryotes that can do photosynthesis, and the chemical reactions are the same. They just occur in a different place. And the truth of the matter is that the organelle that is the chloroplast now was, once upon a time, a prokaryotic organism of its own. And so something as similar to this guy started living inside of a eukaryotic organism, started losing some of its own traits because it didn't need it anymore, and through the course of evolution has become the chloroplast in the process of endosymbiosis. So within the chloroplast, there are two important structural components. The first is the thylakoid. They look like green stacks of pancakes. And the thylakoids possess the pigment chlorophyll. Chlorophyll. And there's chlorophyll A and chlorophyll B. Same idea, they're pigments. And they're green. That's what makes the plant green. There are other pigments involved in photosynthesis. Some of them are red. But we talk about chlorophyll most. So we'll focus on chlorophyll for now. I'll probably discuss some of the differences in the other pigments somewhere in the photosynthesis because that kind of stuff is very interesting. So I can't help but like get distracted by it, right? So the thylakoids are these green pancake stacks, right, on the ground. The space between the thylakoids is called the stroma. It's fluid filled. And I imagine this kind of like a little city of skyscrapers where the thylakoids make up the buildings and the stromas are the streets in between the buildings and all the air above those streets. So this is the anatomy of the organelle that we are working within. The important parts, the thylakoid and the stroma. Okay. Chloroplast. So now on to the basic chemistry of photosynthesis. Okay. On the board, I have the overall reaction for the process, where I have 6 carbon dioxides plus 6 waters in the presence of the light energy yields the sugar, glucose, and 6 oxygen molecules, 6 oxygen gas molecules. And so we have carbon dioxide and water go into photosynthesis, and out comes sugar and oxygen gas. Right? This is a storyline we should all be familiar with. I'm sure we all get this, right? So the plants take these in and push those up. What I'd like to focus on in this basic overview is how the idea of entropy that we talked about in the last video ties in to the storyline of storing energy in this photosynthetic reaction. And so if we look at the reactants, the things going in, the reactants, we have carbon dioxide, CO2, which is a simple molecule, not much in the way of order or complexity required here. And so I have low entropy here. I have water, which is equally simple. No complexity here. Low entropy here. So going in, I just have low entropy stuff. On the other side, I have oxygen gas coming out, right? Even smaller than those guys. So this is also a tiny molecule, also low entropy. But I have this sugar as well. And this sugar is no longer a simple molecule. It's a complex molecule with a fair amount of organization required. And so, compared to all the other guys, this is a big molecule. So this has a relatively high entropy level. Nope, a relatively low entropy level. Right? And so, in order to build this more complex sugar, we needed to use the energy from the light that the sun was smashing down on this plant. So we went from a low energy state, invested energy, stored it in a high energy state, right? And then it had some trash as well. So these products are an energy storage mechanism through a change in entropy. And so this is entropically disfavorable. So I had to put energy in, and a byproduct of trashy oxygen. Okay? So now I've taken the light energy and effectively stored it in the sugar by decreasing the amount of entropy in the overall system here. If I look at this as a whole, my entropy has gone down, which means I invested energy. Okay? So that's what I'm doing with photosynthesis. I've decreased the entropy. I've stored that energy into this system. So now tying the basic chemical reaction with the basic anatomy, we have this model. We've shifted anatomical model a little bit. So this is obviously not a realistic depiction of a chloroplast. This whole thing would be filled with these thylakoid stacks. For the sake of this demonstration, I'm having one thylakoid stack, and I'm having an area of stroma. We have our reactants that are going into this process. So I have my acute sun blasting light into the region. I have water going in, and I have carbon dioxide going in. Down here, we have the products, the things coming out. So we have the oxygen coming out, and we have the sugar coming out. Now, you'll notice that there are two distinct regions here. I have this side, and I have this side, because there are two distinct sets of reactions occurring. So the first reaction set occurs within the thylakoid. It uses the light energy from the sun. It is called the light reactions. And so this is the set of reactions that are actually harnessing that energy from the light and then producing a midway molecule to hold that energy. And we are going to use that midway molecule to ultimately produce sugar in the end. But the light reactions occur within the thylakoid. They generate that midway molecule. It's called NADPH. It's an electron-carrying molecule. It effectively moves energy from one stage to the next. It also produces a little bit of ATP. It passes the NADPH and the ATP out of the thylakoid and into the stroma region. Right? All that space in between the thylakoids. Within the stroma, the second set of reactions occur. And the second set of reactions is called the Calvin cycle. And so the Calvin cycle utilizes the energy stored in the ATP and the NADPH to create these complex sugars. So it's using that energy to decrease the amount of entropy in this overall system by making these complex sugars. And it's not an easy job. It doesn't happen in one step. And so moving forward in this video, I'm going to describe the steps of each of these processes, but we need to see the overall effect before we can worry about the little minutiae details. So next slide is going to be about the light reactions. So here we're going to look at some of the details about the reactions occurring within the light reactions, the steps that are actually harnessing the energy from the light that is coming in and smashing these plants. And so the anatomical structure of the thylakoid becomes very important here. We have to understand that the chlorophyll is embedded in the membrane of the thylakoid folds. And so having all of these folds allows it to have more chlorophyll, and it allows a membrane space for these reactions to occur. So the increase in surface area is very important here. And so we have the chlorophyll embedded in here actually harnessing the sunlight. And so chlorophyll as a molecule is a very complex structure. And the shape of the molecule itself, right, resonates with a certain frequency of light. And so the chlorophyll shape, when the white light of the sun comes and hits it, right, all those different wavelengths of different vibrational levels of energy, right, carried in packets of photons. And when that photon hits the chlorophyll, it hits it in such a way that one of the electrons within the chlorophyll structure gets excited, right? It gains the energy of that photon that was in that particular spectrum, right? So that spectrum of energy is being absorbed by this electron, and that electron becomes excited, so it has more energy. Ooh, okay? And so it's full of excited electrons here when it's getting smashed with the light energy. There are two what are called photosystems in this whole process. Okay? So we have photosystem 2, which occurs before photosystem 1. I did not write photosystem 1. Let me write photosystem 1. Happens prior to photosystem 1. And so this is the story right here. This is where the chlorophyll is. The light comes in, hits the chlorophyll, and so this is happening here. It excites that electron, so that energy is now stored in this electron. That electron that's now excited is going to move along the membrane of the thylakoid through what's called an electron transport chain. So in the membrane, there are specific proteins, and their job is to pass this high energy electron onwards to the next step, which is going to be photosystem 1. However, along the way, as this electron is passed from protein to protein in this electron transport chain, some of that energy from that extra energy electron, that high energy electron, is being utilized for a couple other processes, right? And so this is where the water within photosynthesis comes in. Okay? This process that we're doing requires some hydrogen pluses, some protons, in order to operate. And it's getting those protons from the water. So the H2O is being ripped up to get those protons out. That leaves me oxygen left over as trash, right? And so the H's are getting involved in these processes, and that trash oxygen is just getting spit out. And this becomes the oxygen that we breathe, or the oxygen the plant is going to use for its own cell respiration in the next video. The H pluses are moving from one side of the membrane to the other, and they are forming a concentration gradient of protons. Okay? So the buildup of protons on one side of the membrane. And this gradient is used to drive the production of an ATP. And now this phosphorylation of ATP through this proton gradient and the electron transport chain is something that we will focus on in detail in cellular respiration. So you don't need to worry too much about the actual mechanism here. What I want you to see is that this is where the water is being ripped up, the oxygen is coming from. So this is one of the reactants, and this is one of the products. It's happening in this stage. So an ATP is popped out, right? So that's holding some energy from that sunlight transfer. And that high energy electron, what's left of the energy, is then given to photosystem 1. So that energy ends up in photosystem 1. Photosystem 1 also has chlorophyll in it. Okay? It's a slightly different chlorophyll, so it catches a slightly different range of color. Right? And then the combination of that electron energy from photosystem 2 plus that new sunlight energy really smashes one of the electrons within photosystem 1, and it boots it up to a very high energy level. Okay? So now I have this very high energy electron. It has enough energy to create the electron carrying molecule NADPH. So this product right here is very, very important, and that is the end game of the light reactions, is to make this NADPH. And it required a lot of energy, and so I got that energy by catching the sunlight, moving it through this electron transport chain, catching more sunlight, and then exciting one of my electrons to the point where it had enough energy to create this NADPH molecule. So all of that energy transfer is now stored in this NADPH, which is what this whole system is going to use to make the sugar in the next step. I think that's everything about the light reactions I wanted to say. Do I want to say it now? Don't want to say it. I'll say it. So, photosystem 1 is called photosystem 1 because it evolved before photosystem 2. Some primitive organisms, some primitive photosynthetic organisms only have photosystem 1. And it works. It's okay. It's not great. It's okay. There is no organism that only has photosystem 2. Photosystem 2 does not work on its own. So that's why photosystem 2, chronologically in the steps, comes before photosystem 1, but in the course of evolution, photosystem 1 evolved first. So this just makes it more efficient. Without photosystem 2, photosystem 1 has to excite itself and then give some of its energy back and then re-excite itself again. And so it's not as good. Okay? Onwards and upwards to the Calvin cycle. So, on to the second step of photosynthesis, the Calvin cycle. Again, this is occurring within the stroma of the chloroplast, so it's in the space between the thylakoids. So we just dumped out NADPH and ATP from the light reactions into the stroma, and they are being used in this process here. So this whole process starts at the top. And now this is where our carbon dioxide is going to come in. So our carbon dioxide enters into the stroma, and it is ripped apart. We're using carbon dioxide as a source of carbon. So the carbon is taken. Technically, I'm going to be using 3 carbon dioxides. And it is being combined with a molecule that's floating around in the stroma already. This IUBP molecule. It is a 5-carbon molecule with some phosphates on it. So when you combine the carbon from carbon dioxide with the IUBP molecule, we get a 6-carbon molecule. And so the first step is to go from being this to this. Now this is not a comfortable molecule. It does not like being this. And very quickly, it will split up into 2 3-carbon molecules. This is a 6-carbon molecule. There's 3 of them because we had 3 carbon dioxides come in. And we had 3 Rubis thingies come in. And it splits up into 2 mirror images with 3 carbons each and a phosphate on each end. Some of the ATP that came from the light reactions is going to be used to phosphorylate each of these. It's just going to shove a phosphate on the backside. I've, in this point in time, decreased my entropy by increasing the complexity of this, right? So I've stored a little bit of energy in here. I've made this a less comfortable molecule through the process of phosphorylation. So I now have 6 of these double phosphorylated molecules. And it's not a comfortable thing, so it's looking to become something else. The NADPH, that high energy electron carrying molecule from the light reactions, comes into play now. It interacts with this uncomfortable molecule, and it becomes the product that we care about. So this is using the energy to become this thing. It's another 3-carbon molecule with a phosphate on the end, but structurally some of the functional groups attached are different. And so I've added these demonstrated functional groups. I have a double bonded oxygen, and I have a single bonded oxygen. Just to differentiate that this is different from this. This has functional groups that we need in order to make our product. The abbreviation for the name of this molecule is G3P. And what this molecule is the precursor to building a sugar. So this G3P is the end game of our photosynthesis process. This is what we're using to make the sugar in the end. And so up to this point, I make 6 of these G3Ps with this process. So I got 6 of these, but I can only afford to dump out 1. So every time I go through this, I make 1 G3P. And the G3P can be combined with other G3Ps to make sugars such as glucose. The 5 remaining G3Ps are phosphorylated within ATPs, and they recycle back into being this starting product, and they go back to floating around the stroma so they can sky over and over again. So every time we go through this, we basically make 1 extra G3P, and we dump that out as a product, and then recycle everything back. Okay? So the energy that was harnessed by the chlorophyll is ultimately stored in this G3P, which is used to make our sugars, which are complex structures, right, with a lot of organization that hold a lot of energy. And so that was the story of photosynthesis. The chloroplast, with its chlorophyll, has caught the energy possessed by the photons of the light. That energy is used to excite some electrons. Those electrons transfer that energy into an electron-carrying molecule in the steps of the light reaction occurring within the thylakoid. That electron-carrying molecule is NADPH. The production of that NADPH also created some ATP. In the process of making that ATP, we required a hydrogen source, so we tore apart water. The resulting oxygen trash is dumped out as oxygen gas. And the ATP and NADPH move into the Calvin cycle, where that energy is used to convert a carbon molecule base through several steps of changing shapes. And so we're going from these comfortable stages and forcing them into uncomfortable stages. And then those uncomfortable stages are finding their way back down into comfortable stages. And so we have shifted through the combination of exergonic and endergonic reactions to create this G3P sugar precursor, which is a relatively organized molecule. We shove G3Ps together to become the sugar. The sugar is a highly organized molecule. And so we went from having a raw reactant stages of having high entropy to having our product with low entropy. In order to do this, we needed to invest energy. So the amount of free energy available for work is lower over here than it is over here. But that free energy is now being held within the mechanisms of organization here, the bonds of this molecule. So we've taken the energy available in sunlight, we've converted it through chemical reactions, and it is now being stored within the bonds of this sugar. And so by manipulating entropy changes, we have effectively saved some available free energy for use when we want to use it. And the way we get that out is through the process of cellular respiration, which I'll speak of in the next video. So that's it for this one. I hope you have a fantastic day. Bye.